Okay, so today we're going to be talking about point mutations.
A point mutation occurs when one base in the DNA sequence is substituted with another. Our bases are thymine and cytosine as our pyrimidines, and adenine and guanine as our purines. There's two different types of substitution: a transition, which can be a purine to a purine, like A to a G or a G to an A, or a pyrimidine to a pyrimidine, like a T to a C or a C to a T. And then we have transversions, which is a purine to a pyrimidine, or pyrimidine to a purine. So we could go, for example, from adenine to cytosine, or thymine to guanine. There's 4 different point mutations. There's a nonsense, which occurs when the substituted base turns the codon into a stop codon. A missense, when a substituted base changes the amino acid that's formed, so now a new amino acid is formed. There's a silent, it's when a substituted base doesn't change the amino acid form, so the amino acid stays the same. And then there's a readthrough. The substituted base changes the codon from a stop codon to one that codes for an amino acid. Okay, so now we're going to do some examples. First, we're going to do an example of a missense. So here's our normal coding strand of the DNA. And the normal mRNA would be methionine, tryptophan, asparagine, alanine, cysteine, and a stop codon. But we're going to do a transversion, changing the A to a C, or going from a purine to a pyrimidine. So you can see here, this is the normal DNA, and here's the mutant DNA with a C there. And so the mutant mRNA has a C there as well. So the change from the normal sequence to methionine, tryptophan, and then a threonine instead of a asparagine. So the change in the base from A to C changed the coded amino acid from asparagine to threonine. Now we're going to do an example of a silent mutation. So that's when it doesn't change the amino acid form. So you can see here, this was the same normal sequence, and it normally has a T here, and we change it to a C, which is a pyrimidine to a pyrimidine, so that's a transition. And it didn't change the fact that alanine was formed here. So the change from T to C didn't change the amino acid coded for at all. Here's an example of a readthrough. A readthrough is when the stop codon is changed into one that codes for an amino acid, so it doesn't know to stop and it keeps coding on. So you can see here, we did a transversion from A to C, a purine to a pyrimidine. Because normally there was an A in the normal DNA here, in the mutant DNA, we changed it to a C. So it caused the mRNA to be mutant and have a cysteine here instead of the stop codon that it normally had for UGA. It's now UGC, the cysteine. So it changed it from a stop codon, and it continued to read through because there was no stop here anymore. And so proline and glycine were also coded for. And lastly, we're going to do an example of a nonsense. So a nonsense is when a normal codon that codes for an amino acid is changed to a stop codon. So normally here we had tryptophan, and we did a transition changing from G to A, which is a purine to a purine. See here, the normal had G, and now the mutant has an A. So instead of being UGG, which is tryptophan, it's UGA, which is the stop codon. So none of these got coded for because it stopped here. Thanks for watching.